The anatomy of the cerebellum. The cerebellum is situated in the posterior cranial fossa and is covered by the tentorium cerebelli. It lies posterior to the fourth ventricle, the pons, and the medulla. It consists of two cerebellar hemispheres joined by a median vermis. The cerebellum is connected to the brain stem by the superior, middle, and inferior cerebellar peduncles. The cerebellum is divided into three main lobes: the anterior lobe, middle lobe, and the flocculonodular lobe. The anterior lobe is seen on the superior surface and is separated from the middle lobe by a V-shaped fissure which we call the primary fissure. The middle lobe is also called the posterior lobe. This is the largest part of the cerebellum and it's situated between the primary and the uvulonodular fissures. The flocculonodular lobe is situated posterior to the uvulonodular fissure. The cerebellum is composed of an outer covering of gray matter called the cortex and an inner white matter. Inside the white matter, there are masses of gray matter which form the intracerebellar nuclei. The structure of the cerebellar cortex. The cut surface has a branched appearance called the arbor vitae. The gray matter of the cortex has three layers: the outer layer, the molecular layer, the middle layer called the Purkinje cell layer, and the internal layer, which is the granular layer. The molecular layer contains two types of neurons: the outer stellate cell and the inner basket cell. There are neuroglial cells found in between. The granular layer has small cells with densely staining nuclei and scanty cytoplasm. Each cell gives rise to 4 or 5 dendrites which synapse with mossy fibers. The axon of each granule cell passes into the molecular layer where bifurcates at a T-junction. These fibers are called parallel fibers and they run at right angles to the dendritic processes of the Purkinje cells. Golgi cells are also present in the granular layer. There are intracerebellar nuclei within the white matter, and these from lateral to medial are dentate, emboliform, globose, and fastigial nuclei. The white matter. There is a small amount of white matter in the vermis, which closely resembles the trunk and branches of a tree, and therefore it's called arbor vitae. The white matter is composed of three groups of fibers: intrinsic, afferent, and efferent. The intrinsic fibers do not leave the cerebellum but connect to different regions within the organ. Afferent fibers form the greater part of the white matter and proceed to the cerebellar cortex. The efferent fibers start as axons of the Purkinje cells of the cerebellar cortex. Most Purkinje cell axons synapse with the neurons of the intracerebellar nuclei. The axons of these neurons then leave the cerebellum. The cerebellar cortical mechanisms. The climbing and the mossy fibers are excitatory to the Purkinje cells. The climbing fibers are the terminal parts of the olivocerebellar tracts. The mossy fibers are the terminal fibers of all the other cerebellar afferent tracts. The Purkinje cells form the center of a functional unit of the cerebellar cortex. The cerebellar afferent fibers from the cerebral cortex. The cerebral cortex sends information to the cerebellum by three pathways: the cortico-pontocerebellar pathway, from which you have axons arising from nerve cells in the frontal, parietal, temporal, and occipital lobes, and they go down through the coronal radiator and the internal capsule and end on the pontine nuclei. The pontine nuclei give rise to transverse fibers of the pons. These cross the midline, enter the opposite cerebellar hemisphere as the middle cerebellar peduncle. The second is the cerebro-olivo-cerebellar pathway. Fibers arise from the nerve cells in the frontal, parietal, temporal, and occipital lobes of the cerebral cortex, and they descend through the corona, radiate an internal capsule to terminate bilaterally on the inferior olivary nuclei. The inferior olivary nuclei give rise to fibers that cross the midline and enter the opposite cerebellar peduncle. Cerebro-reticulo-cerebellar pathway. The fibers arise from nerve cells from many areas of the cerebral cortex, and they terminate in the reticular formation on the same side and on the opposite side in the pons and medulla. The fibers from here enter the cerebellar hemisphere on the same side through the inferior and middle cerebellar peduncles. Cerebellar afferents come from the spinal cord as well through three major pathways, which are the anterior spinocerebellar tract, the posterior spinocerebellar tract, and the cuneo-cerebellar tract. The cerebellar afferents come from the vestibular nerve as well. The vestibular nerve sends many afferent fibers directly to the cerebellum through the inferior cerebellar peduncle on the same side. All the afferent fibers from the inner ear terminate as mossy fibers in the flocculonodular lobe of the cerebellum. Cerebellar efferent fibers. The entire output of the cerebellar cortex is through the axons of the Purkinje cells. Most of the axons of the Purkinje cells end by synapsing on the neurons of the deep cerebellar nuclei. The axons of these neurons form the efferent outflow from the cerebellum. These are 4 in number: globose emboliform ruberal pathway, where the fibers go from the globose and emboliform nuclei to the red nucleus, and from there the rubrospinal tract. You have the dentothalamic pathway, where fibers go from the dentate nucleus to the ventrolateral nucleus of the thalamus. You have the fastigial vestibular pathway, where fibers go from the fastigial nucleus to the lateral vestibular nucleus, and from there the vestibulospinal tract. Then you have the fastigial reticular pathway, in which fibers go from the fastigial nucleus to the reticular formation, and from there to the reticulospinal tract.